Welcome to today's live stream. My name is Aiden. I am an MCAT tutor. I scored a 527 on the MCAT exam when I took it last year. And this year, I am starting medical school at the Albert Einstein College of Medicine in New York. And I'm starting school in August. Before I start school and while I'm in school, I am going to be tutoring for the MCAT. I have had 5 students that I have successfully tutored to take this test so far, and I welcome you to join them in being my student. And in order to do that, you would go to aidenmcat.com, and the first step would be to book me for a free online consultation during which we can talk about your preparation as it stands right now and how I can best help you as an MCAT tutor. I love to tutor and I love to teach. And part of that for me is doing these MCAT content live streams where I break open a part of the Association of American Medical Colleges uh, content outline for the MCAT and see what exactly they want you to know about certain topics. So, the topic that we are going to dive into today is covered by Content Category 1C in the AAMC's content outline for the MCAT. So, Content Category 1C is the transmission of genetic information from the gene to the protein. And while other aspects of genetics, such as the nature of the heritable molecule, deoxyribonucleic acid, are covered in other content categories that are outlined by the AAMC, this category focuses on the subtopics evidence that DNA is the genetic material, Mendelian concepts, so Mendel's laws, things like the alleles and homozygous versus heterozygous. It also covers meiosis and other factors affecting genetic variability. So, what are certain things that are going to cause new organisms, new cells to be different than the ones that came before them for the purposes of genetic diversity and thus evolution? And finally, analytic methods, which includes the Hardy-Weinberg principle, gene mapping, and biometry. And finally, evolution. So these are the topics that will be covered in this live stream today. Now, the format of my live streams are that I share a document that I've prepared that is a blank outline. And that blank outline is directly copied from the AAMC's content outline for the MCAT. And on that outline, I am going to type in notes that I take as I discuss these different topics. For this topic, since it's one that I don't often teach, I am going to rely on notes that I find in real time and specifically figures that I find in real time to explain the concepts that I'm diving into today. Because I think these concepts are best explained visually, and I'm also not very brushed up on these specific ones myself. They're what we would call less high yield for the MCAT or less commonly tested. However, as many MCAT practicers would know, those not high yield content categories are sometimes the ones that can be your downfall if you don't prepare for them anyway. So today we're going to dive into this one. It was never my favorite in biology class too, so I guess that also sort of makes this one a little bit more challenging for me. So I have a link to this Google Doc, a public link that anyone can click onto in the description of today's live stream. And I have the document on my laptop right now open. I'm going to share that window of my laptop, and we are going to run through these content points one by one and describe them with figures or images that describe the content. All right, so this is Content Category 1C, the transmission of heritable information from generation to generation and the processes that increase genetic diversity. So the first topic that the AAMC wants us to know is this one, evidence that DNA is the genetic material. So we begin this discussion with the assumption the assumption that deoxyribonucleic acid is that molecule that will pass traits down from one cell to the next. That's an assumption that is based on many studies. And a few of those studies, the MCAT wants you to know about. So why don't we now go and find which one specifically and learn a little bit more about the protocol of those studies. Okay, so I just took a screenshot from a common third-party study aid called the Miles Down review sheet, and I will go through the outline that they give and also try to pull in information from other sources. So the first experiment that the AAMC likely wants us to know is the Griffith experiment. So transformation is the process by which bacterial cells can take DNA from their surrounding environment and incorporate that DNA into their genome. It's one way that bacterial cells have of diversifying and of becoming so well adaptive to their new environments. So transformation. Well, what makes transformation evidence that DNA is a heritable molecule, one that will instruct the next generation of cell, of living thing, to be what it is? Well, in the Griffith experiment, okay, so here is a picture of the Griffith experiment. Crop this image. Let's save the rest for later. We're going to start with Griffith. Okay. So in the Griffith experiment, it seems like they injected these mice with different strains of virus, different strains of bacteria. It's important to note that these are bacterial cells that are causing either the death or not death of these mice. So there's the rough strain, which is virulent, meaning it does not kill the mice. And then there's the smooth strain that is virulent. Okay. So it seems like when you take the rough strain and you inject it into the mouse lives. That's because this strain of bacteria does not possess the properties necessary to kill the mice. When you inject the smooth strain, it's the opposite, that the mice die, and that means that it's virulent. But what about when you take the smooth strain, you heat it up, and you kill the bacterial cells? The smooth strain, the bacteria, you heat it up, and it dies. Well, if you inject that into the mouse lives, and that's because you didn't inject it with a living bacterium. You injected it with a dead fragments of bacteria that you just killed with heat. But if you take that same heat-killed smooth bacterial strain and mix it up with some rough strain, the mouse dies. So some living rough strain bacteria and dead smooth strain bacteria, the mouse dies. Well, one way that this could happen, which alongside all the other experiments is what we believe to be the way, is that the rough strain bacteria, which originally did not contain the properties necessary to kill the mice, picked up or transformed some of that smooth strain DNA that had been left around after the smooth strain was killed with heat, picked up some of it, incorporated it into its own genome, thereby gaining the properties necessary to kill the mice, and then it killed the mice. So here what is happening in the Griffith experiment is the rough strain bacteria gains the properties of the smooth strain bacteria by gaining its DNA. Um, the next experiment for evidence that DNA is the heritable material. Let's now crop this and reveal it. Is the Avery McLeod-McCarty experiment. Okay. So this is where degradation of DNA led to a cessation of bacterial transformation. Degradation of proteins did not. Okay, so transformation, as up here in the Griffith experiment, is that process by which bacteria will take DNA into their own genome, and they will take that bacteria from the surrounding environment. All right, let's get a similar diagram for this experiment, and we can sort of okay. So here's the Avery McLeod-McCarty experiment. So let's take. I'm going to take a look at this diagram first, sort of learn it, and then explain it to you. All right, let's use a different diagram because that one was actually seems to have been a clip from a video. This is maybe a better, let's see. Okay. So here you start with these heat-killed S cells. Okay, so the S cells are a kind of bacterial cell that Avery et al. Killed with heat at the beginning of this experiment. And since the goal of this experiment is to determine, well, which biomolecule among lipids, sugars, or nucleic acids is it that contains the heritable genetic information? Which biomolecule is it? They started by uh, breaking down basically the cells into their component biomolecules. So if we're trying to see what it is inside that cell that needs to be passed down to the next generation in order to have a new kind of cell that's exactly the same, you got to break that cell open and take everything out of it that was in it before. They do that by heating up the cells, and the cells in this experiment were called the S cells. Okay, so once they heat up those cells, first they take out the lipids and the sugars. So now we're dealing with proteins and nucleic acids, I guess, because these are the main classes of biomolecules, the four of them: nucleic acids, sugars, lipids, and proteins. And if you take out the lipids and the sugars, we're primarily now studying protein and nucleic acids. Well, there's three conditions after that, which correspond to proteins as the broad category, and then two classes of nucleic acids, which are the most commonly talked about, deoxyribonucleic acid and ribonucleic acid, so DNA and RNA. So what Avery et al. Proceeded to do was take what was in those heat-killed S cells, remove either the protein, the RNA, or the DNA, and then see if those S cells could then go on to proliferate via transformation by the R cells. So the R cells are this other bacterial strain that you put in, and just like in this experiment, just like with that rough strain, that non-virulent rough strain, you would expect those cells to undergo transformation of the DNA that surrounds them. So here, the R cells are picking up the DNA. Here, the R cells are picking up the DNA. Here, the R cells are not picking up the DNA. Why is that? Because you added this enzyme called DNase, rather, that removed all of the DNA, destroyed it. So here, R cells do not proliferate into S cells. No S cells appear. Here, when you removed the RNA, and added R cells, the R cells did transform the S cell DNA, incorporated it into their genome, and then became S cells. Here, when you removed just the proteins via a proteinase enzyme, this would be an RNase enzyme. When you remove the protein and you put in the R cells, they still undergo transformation and S cells appear. So, what might be going on here then? Well, perhaps DNA is the molecule needed to turn those R cells into S cells. And that would suggest that DNA is functioning as an inheritable set of instructions for the cell to be the kind of cell that it is. Okay, so that's the Avery experiment. So we've done the Griffith experiment, the Avery experiment, and now for the final experiment, I think this is the one with molecular weight. That was a cool one. Let's see. Okay, so we put that here. I'm gonna crop it. Oh yes, the Hershey-Chase experiment. Okay, so the Hershey, so this is again the some third-party study aid that I'm using. Hershey-Chase confirmed DNA is the genetic material because only radio-labeled DNA could be found in bacteriophage infected bacteria. So let's see what that's about by finding a diagram. Okay, so straight out of Wikipedia, this is the diagram for the Hershey-Chase experiment. Let's put it on in the next page. Okay, so bacteriophages are a class of virus, so not a living cell, but a protein complex that contains genetic information in the form of either DNA or RNA. It also contains proteins necessary to turn that genetic information into using the host cell into new virus. Okay, so bacteriophages, they're called that because they sort of eat or phagocytose. Or phage is like an eat sort of. I think it's Greek or something. So phage eats bacteria. Okay, so these are bacteria eating viruses. They're the enemies of bacteria. They destroy them. What Hershey-Chase did is they took certain bacteriophages where they labeled sulfur. And then they took other bacteriophages where they labeled phosphorus. So between sulfur and phosphorus, well, let's take a look at the structure, the molecular structure of DNA. Okay, so here's the molecular structure of DNA. This is a pretty awful diagram, but it does have that atom that I wanted to see. So phosphorus is an element that is part of what's called the phosphate group on the backbone of DNA and RNA. So these P's represent not phosphorus atoms, but phosphate groups, which is a phosphorus with some oxygens and it's negatively charged. But in the middle of that is the phosphorus atom. So atoms can be labeled with in different ways to sort of exhibit these nuclear radioactive phenomena. And we call that radio labeling, where we basically take one atom, incorporate it into a molecule, but that atom is different from normal because it's radioactive in a certain way. It's very useful research wise because you can pinpoint where atoms are going. So in this case, they radio labeled the phosphorus atom. And so that would incorporate itself into DNA, and indeed it did. So if DNA is green, then we see the DNA here. It's in the virus. We see the DNA go into the bacterial cell, and then we see more and more DNA because the DNA stays there. And then after centrifugation, which is just like spinning a tube of the sample that contains all of this stuff, you see phosphorus in the cells. So phosphorus, that radio labeled phosphorus, is still there. How about when you radio label sulfur? So unlike DNA, sulfur does not exist in the backbone of amino acids, which is the monomeric component of proteins. Um, but there is one amino acid, um, or actually two amino acids, cysteine. Okay. So check out these amino acids. These are called cysteine and methionine, and they contain sulfur atoms. So there's the S and there's the S. So what Hershey Chase did is they labeled to find basically where the proteins were going, they labeled sulfur. To find again where the DNA was going, they labeled phosphorus. So I'm showing you now, this is where proteins have their sulfur. It's in methionine and cysteine side chains. This is the side chain of cysteine. This is the side chain of methionine. And with DNA and phosphorus, they have it in the backbone. Okay. Sulfur does not show up in the cells that are infected by the bacteriophages. So that suggests one possibility is that what's happening is that DNA is going from this bacteriophage into the cell and staying there. And that's how viruses pass their genetic information into a cell. Whereas if we had seen protein at the very end, the protein that had started with that virus, it would suggest that that viral transmission of instructions is via proteins. But that doesn't seem to be the case after the Hershey Chase experiment. So altogether, we have, for that first point, evidence that DNA is the genetic material. We have the Griffith experiment, where you turned bacteria that did not used to be deadly into bacteria that is deadly merely by adding some DNA. Okay, so that means maybe DNA is the genetic information. Then you had the Avery McLeod McCarty experiment, where taking away the DNA from these fragments of a killed cell, taking away the DNA causes there to be no uptake of any sort of genetic information by new cells that you add to a solution. And then in the Hershey Chase experiment, you have this special glowing radioactive phosphorus that stays in a cell once it's been infected by a virus. Whereas when you have that glowing radioactive sulfur, it does not stay in the cell after it's been infected by the virus. So together, this is the evidence that the AAMC wants you to know for DNA being the genetic material. It's very important that we re-examine the foundations of science like that. Okay. Now that we have the molecular basis of all of this inheritance stuff out of the way, the next concept that the AAMC wants us to know about is the Mendelian concepts. Okay, so Gregor Mendel is the Austrian biologist behind the Mendelian concepts. So there we go. This is why this topic is called this. Okay, it's because of this man pictured here. Okay, in 1862. All right. So there's a lot of different scientists' work here, but that's why the heading is Mendelian concepts, because it's like this guy. Okay. So the phenotype and the genotype. The difference between these two things is illustrated pretty well by this figure that I prepared before the video. So here, the genotype of an organism is literally its molecular instructions. So if you took any cell in the body, with some exceptions that don't have genetic information in them because they've matured into a state without genetic information, such as like red blood cells, they don't have the nuclei, they're anucleated. Anyway, if you took basically any cell in the body and plucked out its genetic information, you would see that thing on the left. Okay, that thing on the left is the genotype. You know it's talking about the genes because it has gene in the name. So the genotype is the genes themselves. But why are the genes so important? It's because they make a human or any organism what that organism is. So, what's the word for that part? The part where the organism actually shows the results of those instructions. That's the phenotype. So, they're just talking about different things, genotype and phenotype. They're different terms that we got to use to describe what we're talking about. Genotype is the DNA, phenotype is the trait, the thing that appears physically as a result of that DNA. Okay, so here's an example. There's some DNA there, and that DNA either coded for blue eyes or it coded for brown eyes. And then, well, what's going on with these letters? Okay, so let's go to the next topic to figure that out. Well, okay, before we get there, a gene, a gene is it's important to define so that we don't get it mixed up with anything else. The gene is just the part of the chromosome that codes for a certain thing. So a chromosome is just a big bundle of DNA with a bunch of other stuff, but let's just call it a big bundle of DNA. And then part of that chromosome that codes for whatever thing you're talking about, whatever trait that ends up appearing, is the gene. So the gene is that little fragment of the chromosome that's so important because it's going to make something what it is. Okay. Um, but how do we talk about the position of that gene on the chromosome? The chromosome is a physical thing. It's a physical um, bunch of DNA. And if we don't have sort of ways to describe what DNA we're talking about, it can get very confusing because it's really just a big jumble and it looks like a big hairball. So the locus is the location of a thing on a gene. So let's see. Let's find a good locus diagram. Okay. Oh, this one actually comes from an MCAT prep company. Okay, great. Let's use it. So there's a chromosome, and there are gene alleles, which we're about to discuss, and locus is just the part. So here you would say like, oh, say the reference frame, the way that we're describing location is by how far up this physical diagram of the chromosome is from top to bottom. Let's say it's like 60% up. That would be the locus of the gene. Um, an analogy is like the locus of New York City is on the east coast of the United States. That's its locus, its position. Okay. Next is alleles. So let's get a good definition of allele. Okay, so it turns out that we have two copies of every set of instructions on in our body. So everything that makes you you has another copy of it sitting around somewhere, and we refer to those two entities, the one that is the one copy and the second copy as alleles. So rather than just calling them copies, there's this special term called allele. Um, let's see what they mean by single allele versus multiple allele meaning. I don't know what they're getting at with that. Okay. So one possibility is that of alleles in a certain population. Okay, so one example of an allele that's used up here is eye color. So the small b would be an allele, and the big B would be another allele. or another copy. And you can see, since we're talking about humanize, and as I mentioned before, humans have two copies of every set of instructions. There's two alleles per human. But let's talk about, rather than just two humans, let's talk about like a group of humans, a population. Say there's a settlement out there with like 2,000 humans sitting around with these alleles. There could be way more than just two different eye colors, and indeed there are. When you observe, I mean, eyeballs is an example that applies. When you observe different eyeballs in nature, you see that there are many, many possibilities. So multiple alleles for that in the population. Other things might be a little bit more tightly regulated when it comes to the population. Perhaps there are other traits that are so important to survival that there aren't a million different versions of it, that you need things to be a little bit more uniform. So maybe that's what the AMC is getting at. But yeah, alleles are just like the copy, which copy you're talking about, this allele or that allele. Okay. Homozygosity and heterozygosity. So the prefix homo, same, the prefix hetero, different. So let's see, homo versus heterozygote. Okay, this is a great diagram, again from Wikipedia, for homozygote versus heterozygote. So here on the left you have capital B, capital B. This is one allele, that B, one version of the gene, and the locus of that gene is at the bottom left of this leg-like chromosome. And then the other copy of that allele, the other copy of that gene is on this right chromosome over here. So in the human cell, you'll have two of these. They look like this. This is actually like a picture of them. I think this is before any sort of replication that is done to make new cells. But yeah, this is what it looks like. So you literally do have these two copies. Everything that makes you is duplicated. And one is basically from your mom and one is basically from your dad. And that's what you have in most of your cells. Okay. So anyway, there's one allele over here, one allele over here. If they're the same thing and they're the capital B in this example, you call it homozygous. So same homo. Over here, if it's lowercase b and lowercase b, it's also homozygous because it's the same. So same homozygous. But in the middle here, there's two possibilities. Maybe the one from dad is capital B, and maybe the one from dad is lowercase b. And at the same time, the one from mom, if it's dad is capital, is lowercase b. And the one from mom, if dad is lowercase, is capital B. In both of these cases, the chromosome would be heterozygous with respect to that trait. So you would have two different sort of possibilities for that trait. And those possibilities come from the population. So there's a million kinds of B traits, and you just happen to have inherited two different kinds of them. And in this case, you just happen to have inherited the same kind of them. So heterozygous, homozygous. Okay. Wild type. That's an important term to know for the medical college admissions test, specifically when it comes to analyzing passages that include experiments. Because often researchers need to describe what, here we go, what kinds of organisms are using at different parts of the experiment, different parts of the experimental design. So here, okay. Okay, so nothing to be afraid of with these superscripts. That C, capital H, it's just another way of capitalizing. You can't capitalize everything. Or you can't do with just capitalization when you have more than two different alleles that you want to describe. So sometimes you have to have superscript or a subscript or something different. So in this case, capital C C is wild type, and wild type is corresponding to this phenotype here. that results from this genotype, CC, and then all of these are not wild type. So what makes this guy, this brown fur rabbit, different from all these other rabbits? I think these are rabbits with different phenotypes. All right, for this one, we're going straight from the Google search result with help from AI, but this is exactly how it is from what I remember it. So the wild type is the standard, typical form of a gene, trait, or organism as it naturally occurs in a population. So let's just say the wild type is the genetic baseline with respect to a population. There can be variability in the population, but wild type is the, let's see, the standard common one. Okay, so when researchers are describing the things that they do to animals to change their traits, such as playing around with the genome, the reference point that they use is that wild type. And this is even done in introductory biology experiments to understand genetics. You use wild type animals, which means the standard, the stock. You take the animal, and that's the way it is. Okay. And then you do other things. So you can make a chinchilla or a Himalayan or an albino. But remember, the baseline that we're using to refer to all of those other things is wild type. So here the wild type happens to be CC, genotype, brown fur phenotype. That's wild type. Okay. But what about all this capital and lowercase stuff? Well, that brings to mind the concept of recessiveness. So let's go back up here. So there's a capital B allele and a lowercase b allele sort of floating around in the genome. What's going on there? And what is the difference between the capital and the lowercase, and what might be the consequence of inheriting one or the other or both? So it turns out that there's certain alleles, since you have two copies for every trait, there's certain alleles that will show up physically if there's only one copy of them inherited. So usually those are denoted by capitalization. Let's see. So here, for example, let's say that capital B is a dominant allele. That would mean that lowercase b is the recessive allele. So let's say we're talking about eye color. In this case, if capital B corresponds to blue eyes and lowercase b corresponds to brown eyes, then when an organism is heterozygous, meaning that it inherits a capital B and a lowercase b allele, the capital from, let's say, dad and the lowercase from mom, it will express the capital trait. It will express blue eyes. That's the definition of dominance. So the capital B allele, which codes for blue eyes, is in essence dominating the phenotype of the resultant organism. Recessiveness is referring to that other allele, that lowercase b. So in the blue eyes, brown eyes example, the lowercase b, the recessiveness, is referring to brown eyes. So how then does an organism get brown eyes if capital B is so dominant? Well, right here. You get brown eyes if you inherit both a lowercase b and a lowercase b. Those two alleles together will make a brown-eyed organism, if we're talking about eyes. Okay. So that is the concept of recessiveness. But it's not that simple. You don't just get away with knowing dominance and recessiveness because it turns out that in nature, there are these other circumstances, these other forms of dominance. So recessiveness, lowercase b, need to inherit two copies of this allele to have it be expressed. Okay, So that's recessiveness. That's the simple part of things. What about complete dominance? It might be referring to what I think it's referring to. Okay, so complete dominance is exactly what I described before. So capital B masks lowercase b entirely. So with the example of brown eyes and blue eyes, let's see, B equals blue eyes, lowercase b equals brown eyes with complete dominance. B masks B entirely, and a heterozygous organism has blue eyes. Okay. How about this thing called codominance? Well, that's where there's more than one dominant allele. Which in this example would mean that there's this other sort of capital B like allele floating around that will reflect itself in the phenotype. So codominance. Okay. Here's a good diagram for codominance. Okay. So a complete dominance here. This is the superscript is not to be feared. It's just a different way of expressing two different alleles. So C uppercase B and C Y. B is the dominant, Y is the recessive. You make a B or blue fuzzy ball. If you have both the dominant B's, you make a blue fuzzy ball. If you have both the recessive Y's, you make a yellow fuzzy ball. Y for yellow, B for blue. That's complete dominance. Codominance. That they show up in the phenotype. So they're both there, but they're not in. So both in codominance, both the B and the Y are there. They're expressing themselves, but they have their different parts of the body that they're kind of expressing themselves on. And then with incomplete dominance, you get the B, you get the yellow, but when you get them together in the heterozygous genotype, you have an intermediate phenotype that is incomplete dominance. So neither the blue nor the yellow gets its day in the sun. Both of them are there partially. So that makes green. If you mix blue and yellow paint together, you get green paint. So it's almost like that. Okay. Incomplete dominance. Okay. I don't know why the AMC puts incomplete dominance right next to leakage, penetrance, and expressivity, but maybe we'll learn right now as we try to figure out what those terms mean. 1 sec. Right. So these are genetics concepts. That's what Google says. That's great. Great to know. Yes, we're talking about genetics here. Penetrance and expressivity. Let's use this study aid for the MCAT. So penetrance refers to the probability of a gene or trait being expressed. So penetrance is measured by what phenotypes are seen. So the expression. So let's put that here. Penetrance probability that a gene or trait is expressed, and that could be sort of sussed out by the name penetrance or penetration. It's how much that gene or trait penetrates the population in question. That is penetrance. Okay. Let's see. Okay, leakage and expressivity. So continuing on in the study aid, leakage is the gene flow from one species to another, or the introduction of new alleles from an outside source. Um, so, as discussed before, there are various different kinds of alleles for any given gene that's going to be inherited in a population. The flow of those alleles from one group to another is called leakage, and that also can be sussed out by the name, because it's almost like a leaky pipe, like water or genes are coming out of one population and going into another. So leakage is the flow of genes slash other. Okay, and that would increase the genetic diversity of a given species, certainly. Okay, let's call it alleles, right? Because it's not really new genes. It's just new versions of the genes. Versions of the genes are called alleles, and then expressivity. Expressivity. Okay, what do they mean by expressivity? Expressivity determines how well genes are expressed. Okay, let's get a better definition of expressivity because that hardly really captures it. Expressivity generally refers to how strongly or in what varying degrees a trait or characteristic is displayed. And this is another medical school like aid here that's going to give us a visual for expressivity. So here, variable penetrance. The allele itself is not being expressed in half of these eggs. So there's 1, 2, 3, 4, 5 eggs that have the blue, and 1, 2, 3, 4. Okay, so almost half of the eggs are white. They're not expressing the blue, and you could also do it the other way. Half of them are not expressing the white. That is variable penetrance. Penetrance, the probability that a gene or trait is expressed. Okay. Leakage isn't really shown here, but it's the flow of alleles from one population, let's say, to another. And then expressivity is the degree to which the allele is expressed. So here, the expressivity is variable because the blueness of those eggs is variable. So the blueness of those eggs is not always the same. It's not always the same. It varies, and that is variable expressivity. And again, suss it out from the name: expressivity, expression, variable expressivity, not all expressing the same. Okay. Hybridization and viability. Okay, so a hybrid is generally like a mix of different things. And I have a hunch about what they're getting at here, but let's get a good diagram going. Hybridization biology on Google Images. Okay. Boom. Okay, so here is hybridization in plants, and it's shown with a red plant parent and a blue plant parent. The hybrid, which is denoted by the descriptor F1, which refers to like the first generation after you combine the genetic info from the parent generation. That F1 hybrid is purple, so it's a little red and it's a little blue, and having the traits of both of those parents, it is the hybrid. So a mixture, a hybrid. This is another diagram for hybrid that came up on Google Images. Let's see what this says. So sometimes when you form these hybrids, it can increase the fitness of the organism. And there are several ways that that could happen. And then the process by which those organisms that are more fit are passed down their genetic information to the next generation, well, that's evolution. So hybridization is one mechanism by which evolution can occur. Okay, and then the gene pool. I think it's self-explanatory, but let's get a definition of what the gene pool is. Okay. Here's a slide about the gene pool. Okay. Populations are made up of members of the same species that interbreed. Variation naturally occurs among the genes within a population. We've discussed both of those points. The collection of all genes and the variations and the various alternate or allelic forms of those genes within a population is called a gene pool. So all of those possible alleles, the blue eyes, the green eyes, the red eyes, those phenotypes are all based on there being different alleles for eye color. And referring to those alleles together, one is referring to the gene pool because the alleles are the genes themselves. The traits that you see, the eye colors themselves, that's not the gene pool. It's the molecules that result in those eyes being those colors. Okay. So that covered the first major heading of content category 1C, and that heading was or the second major heading, because the first was evidence that DNA is a genetic material. The second was Mendelian concepts. All right, so goodbye to Mendel for now, although I suspect that he's going to come back up pretty soon. All right. Okay. Meiosis and other factors affecting genetic variability. Okay, so the significance of meiosis. So there's two types of cell division that occur in the human body. There's one whereby one cell in the body, which we would call a somatic cell, somatic because it's body based, body cell, um, and then germ cell is haploid. Yeah. Okay. So the somatic cells in the body are like the cheek cells, the epithelial lining of the cheek that is often scraped off in biology classrooms to demonstrate the fact that you can see things under a microscope when you stain them different colors. So cheek cells are an example of somatic cells or body cells. But then there's other kinds of cells called germ cells, and there's really just those two kinds in the body. So the somatic cells, the ones that make up the body, and then the germ cells, the ones that will then pass on to make new humans when combined with the cells from other humans, and that's reproduction. So somatic cells, the body cells, those undergo a process called mitosis. Mitosis. In that process, an exact copy of the cell that you start with, ideally, is produced. In the process of copying the genetic information and replicating the cell, both of which are necessary to undergo division into two new somatic cells, there are things that can increase the variation of the genes, such as mutations. But luckily, we have many mechanisms in our body to prevent that from happening too often, because if it did, certain mutations that increase the proliferation of lines of cells could occur, which would result in cancer of different parts of the body. So, somatic cells undergo basically, ideally, perfect replication of themselves, perfect cloning of themselves. And that's what keeps the body going until the organism dies. That's perfect replication of somatic cells. But what about when it comes to creating new life? Well, that's germ cells, and those only happen. Those are only produced in the reproductive organs of the body. So for humans, it's the reproductive organs. They produce the germ cells. But could it be mitosis that's producing those germ cells? No, because mitosis is a perfect copy of the cell that comes before. So there's got to be this other process that creates a different new kind of cell. Because if you just took the somatic cell and made a perfect copy of it, it would not be equipped to combine with another cell and grow into a new organism. Humans start off as one single cell and then they grow into an organism. You couldn't just take a copy of a body cell and grow that in well, hypothetically, but like you couldn't. That's not what happens in reproduction. You don't just like grow a new human out of a body cell. Yeah, you do it with a germ cell combined with another germ cell from another human through the process of fertilization. Okay, so what is that process that creates those new germ cells? It's listed right here in the title of this content category, meiosis. Okay, and meiosis, unlike mitosis, does introduce a fair bit of genetic variability in the product. And then there are these other factors that create genetic variability that we'll discuss. But let's start with meiosis. Important differences between mitosis and meiosis, that is best seen visually. Let's get a diagram. Okay, beautiful. So here on the left, we have mitosis. On the right, we have meiosis. 2N refers to two copies of the genome. Um, so I'm going to put here this image now. So let's say that this picture comes from like a somatic cell. This is a 2N. This is a 2N cell. Why is that? Because these two chromosomes, or this chromosome, has these two what we call chromatids, which are like those strands that come together to form the chromosome. Those chromatids are, in essence, copies of one another, but they're different. They're different. One comes from the mother, one comes from the father, and so they're a little bit different. But it's two copies, so we call it 2N. There's 23 of these pairs. You can count them: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. I hope I said 23 before. So there's 23 chromosomes, and each chromosome has two copies of every type of trait that you can have in a human being. One of those copies comes from the mother. One of those copies comes from the father. So indeed, when it comes to replicating that kind of cell, we undergo mitosis, and we go from 2N or two copies to 2N or two copies, and that is called diploid. 2N is called diploid. But as I explained with meiosis, you can't combine two end cells together. You have to combine two halves to make a new whole, to make a new human. So that is the process that is happening here. You start with 2N, but then you split that 2N into 1N, and then you split it down further into even more 1N. But the major difference between mitosis and meiosis is that this is a perfect replication, technically, or ideally rather, and this is a division. You're really making less genetic information here than here. There's way more differences than that, and let's get into them. So here's a more detailed diagram of the process of meiosis and mitosis. Okay, so the phases of mitosis and meiosis are they have the same names, except with meiosis, since there are two rounds, um, you just do it twice. So the phases are PMAT: prophase, metaphase, anaphase, and telophase. And with meiosis, it's prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. And then you end up with the germ cells, which are either sperm cells or egg cells. Okay. So, and those are haploid, the daughter cells. All right. So prophase, immediately different. Immediately different, because here you have two you have exactly what we have up here, except you have twice as much of it. So you just make a perfect copy of this, and then you undergo mitosis. So you have one copy here and one copy here. They align at the center. The blue is all dad stuff, the red is all mom stuff, and they split up, and you end up with the same exact genetic information at the end. Half of it dad, half of it mom. Here are the chromatids. They're exactly the same chromatids that are here at the beginning. The beginning looks different from the end because here you just replicated your chromatids to undergo the process of mitosis. That's mitosis. But what's happening on the right here? On the right, you start with the blue and the red, but then you end up with these like this mishmash of blue, red, blue, red. These are genetically different from these. These are different cells that will give rise to a different organism. It's a different set of instructions. So meiosis one, rather than just having two copies of the parental information, you actually have these two pairs. Um, yeah, these two sets of pairs of the sister chromatids, and they're sort of getting together here, and swapping information before they're separated. So this process of getting together or aligning and crossing over is one of the processes by which meiosis results in genetic variation. So let's get a close up of that of the crossing over. Okay, beautiful. So here you have the bivalence, the two sets of duplicated sister chromatids. I'm saying sister chromatids because these chromatids are the same genetic information. It's just two of them. So there's one over here, one over here. These are supposed to be exact replicas of each other. This comes from mom or dad, and this comes from mom or dad. Two exact replicas of each other. So with crossing over, first you have the combination of them. The chromosomes, and this is a huge simplification. This is like a macro phenomenon, but basically you have these two chromosomes getting next to each other. They entangle themselves. Some of the genetic information from one gets onto the other, and that's the crossing over part, and it happens at the chiasmata, which I think the definition, let's see. Okay, so yeah, that's the point of contact, which makes a little X between the two chromosomes here. That's the chiasmata. And then you have the recombinant or jumbled up version of the chromosomes at the end. And I just want to get one more term in there. That's the synaptometal complex. The synaptometal complex. Okay. So, whoa, that is way too complicated. Okay, I'm not going to use that diagram. All right, so check it out. Okay. Okay, let me okay. So zooming in then on the process of crossing over, there's this complex that forms between these two chromosomes, one from one organism, one from the other. And has a special funky name, and it is the synaptometal complex. So the synaptometal complex, that's like that hairy bit between these two chromosomes. And that's what's going to allow them to attach to each other to swap genetic information. So yeah, that's crossing over, and that's by swapping genetic information, you introduce a fair bit of variation in the resulting organism. So that's important differences between meiosis and mitosis. Okay, let's see what the next topic is. Segregation of genes. Okay. Okay. So this is actually back to Mendelian concepts, but now we have this science-y, less 1860s way of looking at it. So independent assortment is, I believe, one of those Mendelian concepts, but let's look at independent assortment through the lens of meiosis. Okay. So independent assortment refers to the event in meiosis whereby alleles assort independently. So in that overall diagram of meiosis, you see that you sort of sort all this genetic information into different places. You start with one big jumble of it, and then you stuff different bits of it into the new cells. Well, what is it? What is the process by which we choose, where certain genetic information, which genetic information goes where? We don't choose. It's nature. It's a lot of random sort of events, random chance, and that's why we have so much variation. So the process whereby those chromosomes line up at in the middle of the cell is independent assortment. So this is a pretty difficult to read but accurate diagram of what's happening. Basically, is that you have one parent here, one parent here. The way that those chromosomes line up in the middle of the cell when they're about to divide during meiosis, it's anybody's guess as to how exactly they're going to line up. And so it's anybody's guess which sort of cells we're going to get at the bottom, and then ultimately which cell fertilizes the cell of the other organism with which this organism is reproducing. Also random, also random. So that's independent assortment. It's that part in meiosis where those alleles are lining up randomly. Linkage. Okay, so is it really random when it comes to every single gene? Um, Not really. It turns out that genes are inherited together, like all the time. So let's define then the term that we use to describe that, which is genetic linkage. Okay. So let's see. Okay, so when it comes to any individual gene that you're talking about, it's really not random whether you inherit a different gene given that you inherited one of those genes. It turns out that it depends on the locus, the location. Where's locus? Here, the locus of that gene on the chromosome. So here in this diagram, we see that gene 1 and gene 2 are not linked because they're really far away on the chromosome. So during this independent assortment and during crossing over, that synaptometal complex and the sharing of genetic information, you will get brand new orderings of gene 1 and gene 2, or you won't inherit gene 1 and gene 2 together. Whereas if they're right next to each other on the chromosome, they're likely to be linked and they're likely to be inherited together. Um, so I think that that's something that this guy was not aware of. I think I don't know if he knew about independent assortment or no, I don't think he knew about genetic linkage. He certainly didn't know about all the epigenetic um, phenomena that exist. Okay, recombination, single crossovers, double crossovers, the synaptometal complex, and the tetrad. Okay, so synaptometal complex describes that, which is depicted above. I'll just post this down there. Synaptometal complex. That's that hairy bit between these two chromosomes that are getting together to undergo crossing over. So single versus double crossing over meaning. Okay. Oh, another MCAT study aid is where we're getting this diagram. Okay, so single crossovers and double crossovers are going to be explained by the same diagram here. It turns out that there's many ways that these two chromosomes can sort of exchange genetic information. One model is like this, where there's one crossover, and one is where there's two crossovers. So the one is called single crossover, the second is called double crossover. Okay. The tetrad, I think I know what that means. Let's just double check. The tetrad. Tetra meaning 4. Can we get... Okay, perfect. I don't like that one. Basically, oh, here we go. All right, here is a diagram about meiosis that features the tetrad. And that's all we were looking for just now. Okay, so tetrad, here we go. So the tetrad is this structure right here. It's when you have two sister chromatids binding together in this way. This is also the tetrad. So these are the same picture. They're describing the same thing. And so in words, the tetrad is the 4-part structure that forms during prophase 1 of meiosis, that consists of 2 chromosomes with the same genetic information, but different sort of variations of that genetic information. All right, sex-linked characteristics. Okay, so those are characteristics, either characteristics of non-pathogenic organisms or characteristics that do lead to like a pathogenic or pathological state. So sex linkage would mean that those characteristics are inherited on the sex chromosomes. So going back up to this diagram, this is a great diagram. These are the chromosomes before replication in a human cell. So there's 23 pairs of them, and this one hanging out over here, that's the sex chromosome. Okay, so here's another picture of that same thing, that I'm going to use to sort of illustrate the... Okay. Oops. Okay. So here's another picture of that same thing, of those chromosomes or that genetic information. Here. Okay. Make it smaller. All right, there we go. Yeah. Okay. So all of these chromosomes are a little bit different. All of these pairs of chromosomes, all 23 of them, but these ones are very different. This is the XY pair of sex chromosomes, and up here, it would have been the XX, and that determines the sex of the organism. But when it comes to, so speaking of sex, it's sex-linked characteristics. If you inherit a trait that's instructed to be produced by DNA contained in one of these chromosomes, it's a sex-linked characteristic. And that's important when it comes to certain diseases because your sex can determine the likelihood of your carrying or presenting that disease state if it's a genetic disease. So if you're inheriting a sex-linked disease, it matters whether your sex is XY or XX. I'm going to leave that there. Okay, now talking about the Y chromosome. The Y chromosome is really small. That means there's fewer genes on it. Okay? Yeah. Sex determination. Let's get a textbook definition for that one. In general, in biology, as Wikipedia states, a sex determination system is a biological system that determines the development of the organism's sex. So here, our sex determination system is whether you inherit the XX, the XY, or some variation upon those two, because there are variations. So sex determination is just the process by which the thing that we call sex is determined by the instructions that your cells inherit from your parents' cells. Okay, that's sex determination. Cytoplasmic and extranuclear inheritance. Okay, so the cytoplasm is one part of the cell diagram. It's like the jelly that everything is happening inside of. Um, it's not an empty void. It's a very active, crowded, busy room, the cytoplasm. And then extranuclear inheritance. So inheritance that occurs outside of the nucleus. So I have a hunch about what this is getting to, but mm. So extranuclear inheritance is the transmission of genes that occur outside of the nucleus. One example of that is this thing called mitochondrial DNA. So it turns out that there's this part of the cell, here we go, called the mitochondria, and it's one of the parts of the cell. It has to be there for many different cell types. Um, the mitochondria. So unlike other parts of a cell that get their instructions directly from the nucleus where this stuff is held, right? This genetic information is held. Um, unlike that, when it comes to mitochondria, let's get a cell diagram just so we can see mitochondria in context of the rest of the cell. Because it's not, the cell doesn't just have mitochondria, it's a bunch of other important stuff. Okay, so here is that mitochondria in the context of a cell. So there's the nucleus, there's that genetic information. Okay, the mitochondria is outside of the nucleus, and unlike all of this other stuff, its rules are not dictated by the nucleus. Its rules are dictated by its own DNA, called mitochondrial DNA. That mitochondrial DNA is inherited from the mother. I'm pretty sure. Let's double check. Right. Mitochondrial diseases are inherited from the mother, not the father. Mitochondria with their mitochondrial DNA are present in the egg cell prior to fertilization by the sperm. So the mitochondrial DNA is sitting in the egg cell, not in the sperm cell, and thus your mitochondria is your mother's mitochondria, or the mitochondria that was in your mother's egg cell when it was fertilized to produce you. Okay, that's that's extranuclear inheritance. Let's see. Yeah, unsure if there's anything else they're getting at there, but all right, let's move on. Mutation. So, in order to produce a body, cells need to replicate themselves. In the process of doing so, the uh, molecular mechanisms that duplicate inheritable information, DNA, those mechanisms make mistakes. One kind of mistake that can be made is, for example, when we're adding new nucleotides to a growing DNA strand during replication, this thing called DNA polymerase, this enzyme, can add the wrong nucleotide or the wrong monomeric unit of the DNA strand. Um, if that happens, you get this thing called a mutation, and mutations happen all the time. But I mean, they're not all bad, and they don't all have any impact at all, actually. So let's get into the different kinds of mutation. Sometimes they are bad, sometimes they lead to cancer. So other things can also impact the DNA sequence of cells that lead to cancer, but mutation is one of them. Okay, so let's get into the types of mutation. So I mentioned before, let's see, random, let's go one by one here. Random mutation. So looking at the random mutation, let's see, random mutation. Okay, so I'm not exactly sure what the AMC is describing when they say random mutation. So maybe we'll come back to that. Translation error. an incorrect sequence of RNA or of proteins, proteins, it would be amino acids, is produced by the RNA. Okay, let's look at translation errors. So a translation error on the MCAT, according to Google, can refer to point mutations in the DNA that lead to incorrect amino acid incorporation, or errors during the biochemical decoding process. Um, so that would be more of what I was referring to here. So translation, like all the body's processes, is imperfect. Sometimes the wrong amino acid or the wrong monomeric unit of a protein can be added during the process of elongating a protein chain. That would be called a translation error. Another kind of translation error. Let's stop there for that one. Transcription error. For one example of that is an incorrect an incorrect nucleotide is added during the process of producing RNA from DNA. So the process by which DNA, the instructions in our cells, instruct our body to do certain things or the cell to do certain things is by copying that DNA into a different form called mRNA. That mRNA is then changed into proteins, basically. Broad overview. That first part of that is called transcription, the copying of the DNA into the RNA. And then the second part of that is the translation, the copying of the RNA into proteins, which then go do things and build stuff. Okay. So the transcription error, I suppose, is when, yeah, transcription itself, the copying of DNA to mRNA is erroneous in some way. Base substitution. So a base substitution is where the genetic change is a genetic change where a single nucleotide in the DNA sequence is replaced by a different base. And so that can result in 3 different kinds of mutation. So I'm going to screenshot this one. Okay. So let's say that you take a strand of DNA and you swap out one nucleotide or one monomeric unit for a different monomeric unit. Suddenly, the DNA is no longer the perfect copy that you wanted. It's now something new. Well, there are a few resulting errors or mutations that can occur. So for base substitution, you can have a silent mutation. So with a silent mutation, you change the bases, but it does not change the resulting amino acid and thus does not change the resulting protein that is produced from that DNA. So a silent mutation is exactly that. It is silent and it produces the same protein in that. You get the same exact thing. Okay. Next, from a base substitution is the missense mutation. So this is where you get a different amino acid. So you wanted one amino acid, you got a different one. Why? Because you swapped a nucleotide in the DNA sequence with a different nucleotide. It's a mutation. It's something going wrong. Okay. And then there's the nonsense mutation. So that's even worse than well, it really all depends on the context, but that could be even worse than missense because in the process by which mRNA codes for amino acids, which creates proteins, a nonsense mutation would stop the process early. So let's say you're in the middle of a protein. It's like a hemoglobin protein, let's say for the purpose of like simplicity, and you made half the hemoglobin. Hemoglobin, that really important carrier of oxygen that brings oxygen from the lungs, from the air that you breathe into your tissues so that you can keep being alive. So let's say in the middle of that process, there was a nonsense mutation or the ribosome encounters a nonsense mutation and suddenly there's a stop codon, well, suddenly you only have half the hemoglobin, half of the molecule, and that's a problem. That cell might die because it didn't produce that protein, hypothetically. Okay, so that is a nonsense mutation. So that's pretty bad. Inversions. Okay, so let's see. So what if we're talking, rather than a single base, we're talking about a whole part of the genetic code? Okay, so in biology, a segment of DNA breaks off, flips end to end, and then reattaches to the chromosome in reverse orientation. Oh no. So yeah, that's a problem. Inversion it looks like this. Base substitution. So yeah, that's a mutation that makes things pretty different. So this is a chromosome before and after the inversion. The part of that chromosome has been inverted, flipped upside down. Bad. Okay. Addition, deletion, that's deletion. So that can change things depending on what kind of addition or deletion we're talking about. And then translocation is, I think I know what it is, but let's check. Translocation. Perfect. Okay. So here we have a chromosome called chromosome 4, and then another chromosome called chromosome 20. So before translocation, chromosome 4 is all green and chromosome 20 is all pink, and then part of the green bit swaps out with part of the pink bit, and yeah, now you have two chromosomes that are different from the ones that you had before translocation. So let's see, when does translocation occur? Okay. So translocation can really happen at any point by being induced by environmental factors. Yikes! But it could also occur during meiosis, and also it can occur during fertilization. So the combination of a sperm cell with an egg cell. So yeah, not good. Translocation. These are all mutations of DNA. Okay, but they are good in that they produce in that they're responsible for evolution, responsible for the variation that allows certain traits to be passed down, human evolution, animal evolution. Okay. And then finally, mispairing. So I think that's pretty self-explanatory, but let's get a definition. Okay. So this would be where nucleotides are incorrectly put next to each other. So the mispairing mutation, let's just put it here. Okay, so usually it's a nucleotide, adenosine, or sorry, adenine, binds to T-thymine. We're talking about nucleic acid, deoxyribonucleic acid here, DNA. This is normal base pairing. This is what happens usually. Anything other than normal base pairing, and that would be a mutation. So here, base pairing, we have the T and A and the C and G. It's more commonly said the A and T and G and C pair with each other. let's say A pairs with C or A pairs with G, that is a mispairing, an incorrect pairing of bases. Okay. Um, an advantageous versus a deleterious mutation. Um, we're so if if one of these events helps an organism survive in its environment and is passed down to future generations, it's an advantageous mutation. If it does the opposite, then it's a deleterious mutation. Um, yeah, I'm gonna go straight from Google search for this one. Yeah, okay. Inborn errors of metabolism. I have no idea what that's referring to. Their next topic is inborn errors of metabolism. Let's Google that. Okay. So those are genetic disorders that disrupt the metabolic pathways. So there's a bunch of metabolic pathways, such as like glycolysis, that one must memorize for the MCAT. I suppose an error in the genetic code that results in an organism being unable to undergo the typical metabolic pathways. I suppose that kind of error would be called an inborn error of metabolism. Okay. Relationship of mutagens to carcinogens. So let's see. So a mutagen is something that will cause an alteration to the genetic material. A carcinogen is a mutagen that will that will cause a cell to turn into cancer, or can cause a cell to turn into cancer. Um, okay, let's see. So here's a diagram. The only one that seems to be specified here by the AMC is the carcinogens. So really important to know, like compounds in cigarettes, when you combust them, they become carcinogens. Those are known carcinogens. Those compounds in those cigarettes, those are mutagens that cause cancer. So they're tumor forming, cancer causing. That's why they're called carcinogens, because they cause cancer. These other ones don't pay attention to. Okay, genetic drift. We're going to grab a definition for that. Okay, so there's a couple of ways that the gene pool or the available wealth of alleles can change. One of those ways is called genetic drift, which is just a random event, quote unquote random, that changes which genes are available. So one example of this is a natural disaster. It's something that comes along and changes which alleles are existent. So let's say you have like 5 members of your species left. 4 of them have blue eyes, 1 of them has brown eyes, and that is because the brown eyes is the only one with the phenotype such that brown eyes can be produced. And then the brown-eyed organism falls off a cliff and dies. That means that now you only have blue eye phenotypes. It turns out that blue eyes were recessive, and so the alleles for blue eyes are the only ones that exist now, because the brown-eyed one fell off the cliff. That's genetic drift. That is now no more brown eyes. Random. Whereas evolution is like genetic drift, except it's not random. It's that it would be something like, oh, the brown eyes are less advantageous because you can't see as well in the sun or something like that. But genetic drift, that's a result of chance or randomness. Okay. Synapsis and crossing over, these are mechanisms for increasing genetic variability. It's part of the process by which our genes are passed down to offspring, luckily that process increases the diversity of the gene pool, because then um, the species can evolve and gain better adaptations to its environment. Okay, so now the AMC gets into analytic methods for genetics. So how do we take this unwieldy concept of the genes that instruct an organism to be what it is? How do we take that concept and sort of put it in the lab and analyze it? So there's various different methods we can use to do that. But we need a couple of like foundational ground rules when we're analyzing genes, because as mentioned a few times now, there's so many different kinds of alleles and so many random events that determine which alleles are which, that it gets very confusing very quickly unless you have a kind of baseline. That baseline is the Hardy-Weinberg principle. So the Hardy-Weinberg principle uh, states that basically the proportion of alleles in a population is going to be constant forever, and it's not truth, but it's a baseline. So if a population meets certain criteria, then the allele frequencies will remain constant, and there's a few of them. Um, Criteria for Hardy-Weinberg equilibrium. Um, we could definitely get a nice diagram for that. Okay. So our baseline ideal population that we're going to use to judge everything else off of when we're analyzing genes is going to be the Hardy-Weinberg population. So that is a population that is very large. There is no emigration or immigration, so it's large, it's isolated. There are no mutations, so there's no crossing over. There's no um, sun damage changing base pairs. There's nothing like that. There's random mating, so organisms mate with each other rather randomly. There's no rhyme or reason to it, and there's no natural selection, so everything survives equally. No natural selection. So those are the 1, 2, 3, 4, 5 conditions or criteria for Hardy-Weinberg equilibrium. So if all of those criteria are met, then the Hardy-Weinberg principle states that the allele frequencies, or the commonness of any certain version of a gene, is going to be the same forever, forever, and ever and ever. It's the baseline that we need in order to do all of this analyzing that we're going to do of different populations. So in doing that analyzing, some of what we do is called a test cross. So this was illustrated a little bit earlier by flowers. So let's take a look then again at those flowers to see what happens when you cross one with another. So parent A and parent B here are crossed with each other. When it comes to flowers, this would be like some sort of fertilization process. If it were animals, you would have those animals breed with each other and then, or sort of fertilize them in a different way and have them produce an offspring. That offspring would be called the F1. So it's F1. Why is it that they're called called F1 generation? It's an interesting question. Oh, okay. So the first filial generation is used in genetics to sort of denote which generation came first. You start crossing these organisms, and suddenly there's a bunch of different generations, and it gets very confusing. So just like with the Hardy-Weinberg principle, we have certain descriptors, certain boundaries, boxes that we put these ideas in. Okay, so the F1 generation is that first generation that comes after the parental generation. The F2 generation is the one that comes after that. So you take F1 and it becomes F2. Let's look at what backcross means. Okay. So a back cross is when you take an unknown genotype organism and you cross it with one that you do know in order to determine what its genotype was. So if you don't know what this recessive BB genotype is that produces this black dog, then you can cross it with a brown dog where you know the genotype, and you can determine from the results of that cross what exactly you started with here. Back cross was also defined when I Googled it as crossing an organism with its parent. Its parent generation. So going back to F1 and mating the organism with F1 generation. So that's a little bit different, but okay. All right. Yeah, so backcross, Google says that it's a genetic breeding technique where a hybrid organism is mated with one of its parents. Okay. All right. So I guess that's one way that you can get that answer to what's the genotype is by doing what's called a back cross. All right. Gene mapping, crossover frequencies. So let's just chunk that into Google. Okay. So gene mapping is the process of determining the specific locations of genes and other genetic markers on a chromosome. So here's a nice image definition of it. So the locus of a gene is its location on the chromosome. Gene mapping then is the process by which we find out which genes are on which parts of the chromosome. And then let's do biometry. Biometry definition, biology. Okay. Okay. Oh, great. So biometry is the application of statistics to biology. So we have biology, we have this natural phenomenon, and we need to apply math to it in order to analyze it in certain ways. The application of that math is called biometry. So it's measurement in relation to biological structure. One biological structure is the genetic makeup of an organism. Okay. That's not the definition we're trying to use here. No, please, not the AI mode. So it's the application of statistical and mathematical methods to analyze biological and health data. Okay, so the final evolution that is specified by final category of topics that's specified by the AMC for content category 1C. That's what we're doing, right? Okay, content category 1C. It's getting long. The transmission of genetic information from the gene to the protein. That final concept is evolution. So natural selection is the mechanism of evolution that's agreed upon right now. Natural selection. So let's get a good, just textbooky definition for natural selection. Okay, here we go. So natural selection is a mechanism of evolution. It ensures that only the traits that help a species survive and reproduce get passed on to future generations. So you have a generation of organisms. Some of those organisms are going to be better at surviving than others. The ones that survive are by definition the ones that can reproduce. Thus, natural selection occurs. You select naturally for certain traits by having the less advantageous traits disappear from the gene pool. That's different from genetic drift that I mentioned before, because it's not random. Those organisms died because they were maladaptive to their environment. They did not die by the result of a random event. So here, genetic drift is the composition of the gene pool changing as a result of chance. Here, natural selection is natural is a bit of a misnomer because it's really not random. It's not sort of all over the place. It's owing to differences in the survivability of those organisms. That's the reason that certain traits get passed down to next generations. Okay, that's natural selection. The fitness concept. Let's see. So fitness, I would say that it's the extent to which an organism can survive in its environment. Yeah. So here it kind of looks like this. Lower fitness, lower reproducibility, higher fitness, higher reproduction happening because those organisms are better at surviving in their environment. They're better at doing what they need to do in their environment. That means that there's going to be more of them, generation to generation. Selection by differential reproduction. I'm not sure what that means. Okay, let's go. Oh, okay. So differential reproduction describes how individuals with better traits survive long enough to reproduce. So differential reproduction, that's seems to just be the kind of core of natural selection. Let's just get another picture in there. Why not? All right. So individuals with higher fitness are more likely to reproduce and pass on their traits to offspring. Okay. So natural selection and group selection. Group selection. Okay. All right. Internet is slowing down pretty significantly here. The next is evolutionary success as an increase in percentage representation in the gene pool of the next generation. That's what's happening here. So you start with 2 bugs. Well, there's more bugs because you need to reproduce, but the brown bugs are going to represent a larger proportion of the gene pool. Their alleles are going to be more represented in the gene pool because they are higher fitness than the green bugs, which are going to be represented less in the gene pool after a few generations. So group selection versus individual selection. Maybe a little bit more in depth than the MCAT is going to test it, but basically you can have organisms that help each other survive. It's not just, oh, this organism is well adapted to its environment and so it survives and it passes on itself. The organisms display altruism, so they help each other. So that would be group selection, whereas individual selection, you have exactly what we described before, where the single individual is the one that reproduces well and goes on to dominate the gene pool. Okay. And so this is actually going to go up here. All right. So speciation. Well, in all of this, we start to have the lifting of certain traits from one group in to the extent that it creates another group, another species entirely. So throughout evolutionary history, all these different species have arisen, but life sort of all comes from the same place. So how is it that we all get these different species is just this happening, this genetic variation happening, and after a while you get different species because of the genetic variation, and different genes produce different things. Those different things, we call them different species. Eventually, when they get different enough, there are a bunch of different categories and a bunch of crossover and then differences, but let's stick to what the AMC is asking us to know here. Okay, so speciation is that process whereby different species are produced. Like one kind of organism becomes 2 different kinds of organisms. So polymorphism. Let's see if milestone has polymorphism. Oh, no, okay, polymorphism. Inbreeding. Members of a a family of organisms, I guess, breeding with each other. So polymorphism is the occurrence of two or more distinct phenotypes within a single population of species. So dogs are a great example of this, I think, because you have dog, which is a species, but not all the same. Ask anyone with a dog that you see on the street. It's usually the first question. Well, what breed is it? They're not asking what species is it. They're asking what sort of appearance variation does that dog have? And so those different lines are um, sort of the clearest example I can think of right now of polymorphism. Another is these parrots or the whatever birds these are. Okay, um, adaptation and specialization. I think that's just a fancy way of asking for a pretty simple definition here, but let's look it up anyway, just to be thorough. Okay, so in adaptation, you have natural selection, where the genetic changes make certain individuals. The genetic changes result in certain individuals being more well adapted to their environment than others. That's adaptation. And then with specialization, you have the adoption of traits that fill a specific kind of niche. And I actually took that definition off of MCAT Reddit, but um, seems to be corroborated by the other sources that are popping up when you Google it. So, adaptation, exactly what it sounds like. It's survival of the fittest. It's what we've been discussing, where an organism with good traits passes those good traits down. Specialization. Well, how do we do speciation? How do we break up a certain line into different species entirely? You have the adaptation of certain traits to fill a niche. One sort of example that comes off of my head is like birds. They have filled that niche of flying. Before those birds were flying, there were other organisms that weren't flying, and eventually, with genetic variation, they gained the ability to fly. So that process of them gaining the ability to fly, filling that niche of flight, is specialization. You're filling a niche. You're becoming a different species entirely. Okay. Inbreeding, breeding within a population. Uh, let's see. Wait, let's see. Let's just get a good. Yikes. Okay. So my review sheet is not for some reason. Okay, great. So we got to go to a different tab now. Okay, so it appears that live Googling is maybe not as viable as one might think in 2026. So, okay, inbreeding. The mating of individuals or organisms that are closely related through common ancestry. Yeah. Okay. I believe that's one of those MCAT topics that's known outside of the context of MCAT study. Outbreeding. I'm guessing that means not inbreeding, but let's look it up. And then bottlenecks and evolutionary time as measured by gradual random changes in the genome. So outbreeding is the matings of organisms that are genetically unrelated. Mating between Okay. The bottlenecks are. Let's see. Okay, so in a bottleneck, the size of a population reduces, like liquid coming out of a bottle. The diameter of the space reduces, and then after the population size is reduced, suddenly you have fewer variations, or most likely you have fewer variations. Um, and with a bottleneck, the survivors of the bottleneck are random. So population shrinks dramatically. All right, and then finally, evolutionary time is measured by gradual random changes in the genome. If you sequence the genome of organisms across evolutionary time, you can see that there are differences in their genomes, and the result, those are the result of accumulation of random mutations. So it's a technique called the molecular clock. So we assume that DNA mutations accumulate at a relatively constant rate over generations. That means that if you take a generation and compare it to generations before, you can see how much time passed. Right. Okay. So evolutionary time is measured by changes in the genome, random changes in the genome. Okay. So just to recap before ending the live stream, today's live stream covered genetics on the MCAT by covering all of content category 1C. So in content category 1C, I'm not just going to read off the different headings we covered. We covered evidence that DNA is the genetic material, Mendelian concepts, meiosis and other factors affecting genetic variability, mutation, genetic drift, synapsis, crossing over, analytical techniques, evolution, and that's it. So we finished on evolution there. Again, hire me if you're interested in having a private tutor for the MCAT. My website is aidenmcat.com. The website is going to be in the description of this video and is also in the channel that this video is going to be posted on. So there's going to be a link to my website there. And thank you for watching.